Today, we will discuss about PCR. The objectives of this video is the introduction to PCR, the requirements, the different stages, that is denaturing, annealing, as well as extending. So today, we will learn about polymerase chain reaction. Polymerase chain reaction is often known as PCR. This has been developed by Mullis, and for this, he was awarded the Nobel Prize in Chemistry. And what does a PCR do? A PCR makes multiple copies of a small section of DNA or a gene. So, PCR is a machine that can make multiple copies of a small piece of DNA or gene. So, by using a PCR, we can generate thousands or even millions of copies of a small piece of DNA. From this diagram, it explains that from a single DNA, multiple DNAs has been developed. That is, from an initial single DNA, this much DNA has been developed at the end of the process. So this whole process is referred as PCR. That is, from this, multiple copies of DNA has been created. So next, we will be discussing about the requirements needed for carrying out a PCR. A PCR needs a DNA template, that is, the DNA that has to be copied. It needs primers. Primers are short stretches of DNA. These binds to the section that has to be copied and initiates the PCR reaction. Next, it is DNA nucleotide bases, also referred as DNTPs. These adds the DNA bases, that is A, C, G as well as T. We also require a Taq polymerase enzyme. What is the Taq polymerase enzyme? We'll discuss later on. We also require buffer. Buffer is needed to create a right condition for the reaction. Next, we will discuss about the main stages in PCR. There are three main stages, that is denaturing, annealing, and extending. In the first stage, denaturing, the temperature is increased. That is, the double-stranded DNA is heated so that we get two single-stranded DNAs. So, this stage is when the double-stranded DNA is heated to obtain two single-stranded DNAs. So next is annealing. Here the temperature is lowered and the DNA primers are allowed to attach to the template. This stage is annealing. Next is extending. Here the temperature is again raised and a new strand of DNA is obtained. This is done with the help of Taq polymerase. This diagram shows the three stages of PCR, that is denaturing, annealing as well as extending. First, a double-stranded DNA is undergone to a denaturing step. In this step, a double-stranded DNA is separated into two single-stranded DNAs. After this step, annealing stage, a primer is attached to these single-stranded DNA. A primer is a short strand of DNA or RNA that is complement to the template DNA. So a small strand of DNA is added so that it attaches to the template DNA. So it is complementary to the template DNA. After this stage is the extending stage. At this stage, these primers are extended using Taq polymerase. So at this stage, using Taq polymerase, these primers are extended. Next, we will discuss the three stages in detail. First is the denaturing step. In this stage, the template DNA is heated up to 94 to 95 degrees Celsius. and at this temperature the hydrogen bonds break and resulting in single strands of DNA. That is, from a double-strand DNA, two single-stranded DNAs are obtained. The two single-stranded DNAs that we obtained acts as a template. These are the templates for the production of new strands of DNA. Throughout the process, the temperature has to be maintained. This usually takes a time period from 15 to 13 seconds. Next is the unloosed stage. In this stage, a temperature of 50 to 65 degrees Celsius is provided. At this temperature, the primers can attach to the single-stranded template DNA. So, at this temperature, the primers attach to the template, and this attachment is provided with hydrogen bonding. So, what is primer? Primer is a single-stranded DNA or RNA that has 20 to 30 bases. So, it is a very small stranded DNA or RNA is called a primer. And these primers are designed complementary to the template strand. These primers act as a starting point for DNA synthesis. There are two strands of DNA, and these strands are complementary to each other. That is, one strand is running from 5' to 3', while the other is running from 3' to 5'. So, there is a need for two primers. One is a forward primer and the other is a reverse primer. This whole process takes 10 to 30 seconds. The next stage is extending. This is the final step of PCR. Here the temperature is increased to 72 degrees Celsius. So at this temperature, a Taq polymerase is added. This Taq polymerase enzyme adds the DNA bases. So at the extending stage, a temperature is raised at a degree of 72 degrees Celsius and a Taq polymerase is added. So what is a Taq polymerase? A Taq polymerase is an enzyme, and this enzyme is taken from a heat-loving bacteria called Thermus aquaticus. So from a bacteria, Thermus aquaticus, we get Taq polymerase. So a Taq polymerase is actually a bacteria which lives in hot springs. So since they live in hot springs, they can tolerate a temperature up to 80 degrees Celsius. The Taq DNA polymerase can withstand a high temperature. It doesn't mean that all the DNA polymerases can withstand this temperature. The DNA polymerase that is isolated from human can only withstand up to 37 degrees Celsius, that is a body temperature. This diagram shows the Thermus aquaticus, that is the bacteria from which Taq polymerase has been isolated. This diagram shows that Thermus aquaticus lives in hot springs. These Thermus aquaticus are able to withstand a high temperature since they live in high temperature. The optimum temperature of extending stage is 72 degrees Celsius, and at this temperature the Taq polymerase can build a complementary strand. That is, at this stage a single-stranded DNA can develop into a double-stranded DNA molecule. This Taq polymerase attaches to the primer, and after it is attached, DNA bases are added. This addition is one by one, and it is always in the direction 5' to 3'. Now, let us summarize the requirement as well as one PCR cycle. In the basic requirements, we require a template strand, that is the DNA sample. We also need a primer, which is a short strand of DNA. We need nucleotides. We also need a Taq polymerase, which is an enzyme that has been isolated from a heat-loving bacteria, Thermus aquaticus. We also need a buffer. These are mixed in a PCR tube. In the PCR cycle, there are three stages, which is denaturing, annealing, and extending. At the first stage, that is at denaturing, a temperature of 95 degrees Celsius is maintained. At this temperature, the strands separate. At the second stage, that is at annealing, a 55 degrees Celsius is maintained. The primers bind to the templates. At the last stage, that is at extending, a synthesis of new strand occurs by a Taq polymerase addition. These three stages, that is denaturing, annealing, and extending, are repeated 20 to 40 times so that we obtain lots of copies. In a short run, a huge number of copies are obtained from a single DNA piece, and this is because the byproduct of the first cycle is the template of the next cycle. In this way, we can generate a huge number of copies from a specific DNA segment. This particular diagram shows that a template DNA, a single copy of a template DNA has undergone three stages, that is denaturation, annealing, and extending. After the end of extension, we received two copies of the template DNA. That is, from a single copy, we got two copies. At the end of second cycle, we got 4 copies, and at the end of third cycle, we got 8 copies. At the end of 30th cycle, we got 10 raised to 9 copies. That is, the products of the first cycle is the template of the second cycle, and the product of the second cycle is the template of third cycle, and goes the cycle. We hope you understood the basics of PCR. For more such videos, do subscribe to our channel. If you like the video, please like, comment, as well as share with your friends.